Hi, I'm going to present you the tranSMART Autism Pilot project. I'm logging in the platform and I have access with the I2B2 interface of tranSMART of the multiple cohorts being integrated. I'm drilling down the Simon Simplex Safari collection where there's 2,760 patients. Per patient, there's 6,000 to 10,000 different variables. And I'm drilling down to the question, did the patient have epilepsy, yes versus no? Where do I want to put the not sure? It's me who is going to define that depending on the type of analysis that I want to do. So I integrated not sure with the no. And then I'm comparing, doing a running live this query, comparing the two subsets. On the left, epilepsy yes versus epilepsy no and not sure. Then I can compare, so I have a comparison of age, sex, and race between those two groups. And I can now look at another variable, for example, deafness. How do the patient with deafness correlate with the patient with epilepsy, yes versus no? And the 6 patients with deafness are all in the group epilepsy no or not sure. So that's why the chi-square test is not significative. If instead I take a continuous variable, for example, waves extremity, that's the patient wave extremity. In this case, it will do a t-test, which is nearly significative between the two groups, epilepsy yes versus epilepsy no. And so I can generate hypotheses. The objective of tranSMART is, as it was described, the integration of all clinical data, biological data into one place, and then to help you to generate the different hypotheses. If instead of taking just phenotypic data, I can look at different advanced workflow with ANOVA, correlation, heatmap, Hierarchy clustering, and scatter plot with linear regression. And now I can, as an independent variable, I'm going to take a subscale of irritability, and the dependent variable, if instead of taking phenotypic data, I'm going to take a gene expression from the Affymetrix U133 platform, drag and drop, high dimension data, I'm going to select one gene or pathway to run the analysis. I'm doing it on the gene APC, aggregated probe. Then I'm running, in this case, R plus bioconductor running live at Boston Children's Hospital to be able to do the correlation between phenotypic data against this gene from the cell files. And the APC gene expression level is present here, and here I have the subscale of phenotypic data, subscale of irritability, where in this case the results are not significative, but if I want, I can download the raw R data with the which is not one file, it's a zip file containing the R program and the data to be able to rerun the analysis on my computer. If I come back now to my patients with epilepsy, yes, and I want to see how many samples on this national cohort do I have in my lab, in my biobank. So what I can do is I can, with the sample explorer, I have the information that 6 samples with 11 aliquots are available in my lab, where I can have multiple information, where I can, with the contact information, if I want to have more information on this sample. And the last element I wanted to show you is the integration we did of all exome sequencing data into tranSMART, where you can drill down from the output of ANOVA that has been integrated, from example, non-synonymous single nucleotide variant using the RefSeq annotation, looking at one gene. So I can take the same way as we did with fine-grained phenotypic data, I can take any of the fine-grained annotation from the variant exomic function to be able to do correlations. And then I look at the phase genotype of all the patients being homozygous. I make the link saying that those are on the same variant, and then I generate the whole exome sequencing statistics. In this case, it will be creating a dashboard. What I can do is underneath integrate the phenotypic data, epilepsy yes versus epilepsy no, or any other element to do fine-grained phenotypic integration with fine-grained omic integration. And in less than 20 seconds, I can see that 36 patients have a non-synonymous single-nucleotide variant on this gene and being homozygote. Thank you. If you want more information, please don't hesitate to contact us.